Hey, welcome everyone, and sorry about the delay getting started today. Today we have as our speaker, PhD student and my technician, TC Jayalath, who is no stranger to the department since he's been in the department for quite a while. He got his master's degree with Susie O'Connor a few years ago, and after that moved over to my program, where he has been looking at different ways to manipulate horticultural lighting and control environment agriculture. TC has an interesting life story. Born in Sri Lanka, and after getting his bachelor's and I believe a master's degree in Sri Lanka, he won the green card lottery, which allowed him to come to the US with a green card. He worked in Atlanta in various hotels for a while, and then made a very strong case to be admitted into our department when he started working with Susie. And now his education in our department is coming to an end. TC, with that, I'll turn it over to you.
Thank you, Mark. All right, I hope everything works now. Sorry for the small slight delay we had. And thanks, Mark, for your nice introduction. So today I'm going to present to you my studies I did for last little bit over 5 years. So all those studies, the title I came up with is related to manipulating horticultural lighting to optimize crop production in controlled environment agriculture. It works. So just want to give you an introduction why this is important. If you're looking at the population growth over time, in early 19th century, there were about 1 billion people live on Earth. And at the moment, we have about 8 billion people living among us. And then it's expected to be over 10 billion in 2060. So therefore, to provide enough food for this growing population, we need to have more resources. For example, amount of land we use for agriculture. So we need to have more lands to produce more food, or we have to increase the production of available lands. So that's why the controlled environment agriculture is helpful. We also call CES in short. So in CES, as you see in the pictures, so it has multiple layers of crops can be grown. So if you grow this crop in a field, that's going to be several acres of crops. So benefit of this is having high yield per unit land area. And also in CES, we have more efficiency of water and fertilizer use, and we have very limited pesticide use. And also these setups can be done in the lands that we commonly cannot do agriculture. So that's the benefits of CEA. We can have more lands and also have more productivity for available land. However, the problem is, as you see in the picture here, there are so many lights being used. So there's a high energy requirement for CEAs, especially for lighting. Energy cost is one of the main issues in CEAs, just followed by the labor cost, and it can be over 30% of the total operational cost of a CEA industry. According to the US Department of Energy, US energy cost for horticultural lighting just can be over $1 billion per year. Therefore, my broad objective of this study, my dissertation, is to exploring ways to optimize the use of horticultural lighting in CEAs to achieve greater energy efficiency and more productivity. From this, I have 5 different studies I'm talking about. First study, I try to identify the benefits of providing consistent daily light levels for greenhouse seedling production. And the second study, I try to understand I try to determine the underlying mechanism responsible for growth differences between faster grown crops and slow grown crops. So that can help to screen faster grown crops for vertical farms. And the third study done to identify if lighting energy in greenhouses can be reduced by considering the excess sunlight that plants receive on sunny days. And the fourth study was done to identify if adding more nitrogen is favorable when plants receive high light levels. So growers try to give high light levels for plants so they can have their plants getting production comes earlier than regular light levels. So, but whether it's beneficial adding more nitrogen. And the last study I did to quantify the effect of supplemental far-red light on greenhouse lettuce production. So far-red light is a light added to regular lights on most of the indoor farms. I will explain you later. There's a lot of benefits on having far-red light on indoor farms, especially leafy greens. So I wanted to see whether there's also benefits in greenhouse settings. So first study, high DLI with adaptive LED lighting control speeds up ornamental seedling growth. So what is DLI? If you're looking at the solar spectrum, and the visible light is in between UV light and infrared light. From this visible light, if you take the spectrum between 400 to 700 nanometer wavelength, that is called photosynthetic active radiation, far range. So if we measure the amount of photon that we're getting from this far range in a square meter of area within a second, that is called photosynthesis photon flux density. It's called, we call normally ppfd level. And also, if you're looking at within 24-day period, and if we plot all these ppfd values every second we get, and then we add them together, that is called DLI, daily light integral. So now the value is moles per square meter per day. So why this DLI matters? Plant growth increase with providing more DLI, providing more light. But having no light, plant can have slow rooting, delay flowering, and slow plant growth, and also ultimately lower yield. Also, by providing more DLI, plants can have tip burn, that is the burning symptoms on growing tissues, young leaves, and foliage burn, and also bolting. Bolting is early flowering before we want, like lettuce, sometimes you see flowers coming up. We don't want flowers on lettuce, right? So just like that, we need to make sure we provide the right DLI for the right plant. Therefore, there's a DLI guideline to produce plants in CEAs. For this study, we are looking at seedlings. So seedlings need about 6 to 10 moles at early stages, and it can be 10 to 15 moles of light per day at later stages. But if you're looking at the sunlight levels we're getting from on day 1 to day 365 within a year. So this graph shows the sunlight levels received on 14 years in Ithaca, New York. So each of if you take one single day, for example, if you take day 200, there are 14 data points for 14 years. So if you take day 200 again, you can see some years we get DLI less than 10, and some years it can be close to 60. So there's a huge variation of sunlight, DLI we get from sunlight. So because of this variation from day to day, growers often use supplemental lights to have a high production in seedling. But however, those supplemental lights are not precisely controlled. So they can be turning off, on and off depending on the sunlight level, like 50% light turn on the light, and the light is go down, 50% light turns on, light turn off when the light is above 50%, sunlight is above 50%. Are we out from soon? We are there. Okay. So that's where this, or some growers do providing minimum like a small dim light level throughout the photo periods in addition to the sunlight without control because of not having enough sunlight on some days. But we wanted to develop a control lighting system called adaptive lighting control system. So this system we want to develop that plant can get the same light level every single day, same DLI every single day regardless of the weather changes. So to explain you how this happens, so we use a quantum sensor it can detect how much sunlight at the canopy level, it can detect the light level at the canopy. And we also use a dimmable LED lighting fixture and a data logger. So for example, let's say there's a cloudy situation and plant only gets 70% of light that plant needs. And that's going to be read by the quantum sensor. That signal goes to the data logger. Data logger do all the calculations. And with that, it just provides just enough light to reach the 100% light at the plant canopy. So this just keeps happening every second. If there's no cloudy, if you get 100% light from the sun, then data logger turns the light completely off. So this in data logger is just do 3 different steps to calculate these light levels at every second at the plant canopy. First, it just as I mentioned you earlier, DLI, you just calculate the amount of DLI I have received so far by adding the PPFD values together. Then it calculates how much DLI needed at the end of the day to reach the target that we have already set, this DLI target we need at the end of the day. So it just calculates that. And then the third step is calculate the PPFD target. That is the target at every second should be at the canopy level by dividing the DLI needed from the amount of second left to the end of the day. It can calculate the PPFD target easy. So therefore, objective of this study to test the ability of the adaptive lighting control system to see whether it can reach specific DLIs we said, and to quantify this effect of control DLIs on seedling growth. For this study, we used two crops, impatiens and wing crop, low light and high light preferred crops. For this study, we had 5 different treatments. The first one was controlled without supplemental light, sunlight only. And the second one, we try to mimic what some growers do, providing dim light level, 100 ppfd of supplemental light throughout the photo period. And the third treatment, we had DLI target of 8 with our adaptive lighting control system. And then we have DLI target 12 and 16 on the other 2 treatments. And these treatments were randomly allocated in 5 different blocks in the greenhouse bench. And as you can see here, we have both vinca and impatient plants in one section of each section. We had both crops. So over here, and we selected the, like as you see now, also we selected 3 different locations. From all the selected locations, we harvested the 16 plants at the most centered 16 plants from each time. The first time we harvested 15, 20, and the third one was harvested 30 days after placing those seedlings under those lighting treatments. And this shows within 1 day during our study how the adaptive lighting control system works. So over here you can see target PPFD, that is what calculated by Jayalath, and the actual PPFD is what is reading by the content sensor. So it can be actual PPFD, can be sunlight, or it can be LED light, or it can be both. So till about 10 o'clock on that day, we have Jayalath had set target DLI as 0. But however, you can see the actual is going up because the sun's rising and these plants get sunlight. At 10 o'clock, it set target about 250. Therefore, the actual goes up as well. So actual light level now reached the 250 at 10. But you can see eventually the actual go higher than the target because sunlight we get more than 250. So on this day, we get about 600 micromoles on sometimes. So because of having higher sunlight, the target eventually go down. Otherwise, we will exceed the DLI target at the end of the day. So the program does all these, is change the target every single second. And after about 3 o'clock on that day, you can see the target and actual was at the same level. So after 3 o'clock, sunlight may be down. So our LED just give the enough light to keep the actual on target level. So at close to midnight, it's probably just be LED light to keep that target. So that is within a day. And now this is I'm showing you each of the treatment, how much DLI they got within those 30 days of their study periods. So if you're looking at the sunlight, you can see sunlight. Some days we get, very first day we get about 3, and then day 4 we get about 2 moles of DLI. And then some days we have DLIs of 8, like day, if you're looking at day 6 or 7, we get DLI of 8 on that day. So that is, as I mentioned, your DLI changes from day to day. This is the variation that we had within that 30-day period. But for the average, we got 5.4 moles throughout our study period. And this is what growers do. They just keep a dim light level on without any controlling. So they are like, because of providing 100 micromoles here, they are like just, their light patterns, DLI pattern also change just like how sunlight is. But average, we got about 8.6 within that 30 days. And this is how our adaptive lighting control system is. These are actual data points on each DLI on each single day. It just looked like a one straight line because they was able to reach the DLI target every single day. For example, if you think that we have a plant that needs 8 moles of light every single day. So think about these days that what growers usually do. On the days I have marked in blue, that's the day that plant doesn't get enough light. And then I have marked in red. That's the days that those lighting provided extra light than what plant needs. That's just wasting energy. So that is why the importance of having a DLI control throughout the growing period. And just like our 8 volt DLI target, other 2 treatments also reach the targets every single day. So I'm showing you data I got from 2 crops. So first one is shoot dry weight, impatient and winker. So I'm looking at the 30-day harvest, which is the last harvest. If you're looking at the DLI treatment with DLI target 16 and 12, the highest light levels, we did not see any difference between 16 versus 12 on shoot dry weight on impatience. But in winker, we saw at higher light levels, shoot dry weight increased. But if you're comparing the DLI 8 control treatment versus 100 micromole, which gave 8.6 average, that both treatment did not have any difference between shoot dry weight on both crops. But I want to point that out. If you're looking at the dry weight we got from sunlight at 30 days, which is the last one, you can see that that dry weight can be obtained at 20 days just by providing any supplemental light. So that's the benefit of providing supplemental light. So if grower needs that dry weight, they can achieve that dry weight 10 days earlier by providing supplemental light. So time is money for growers. If you can harvest your crops early, set it out, then you can have another batch of crops. So that is their money, the money saving. So that is benefit of having supplemental light. And so the same pattern in both crops. We also look at root dry weight because roots are more important for seedlings. And we saw the same pattern as in shoot. Higher light had no difference between 12 and 16 on impatience, and also no difference between 8 versus 100 micromoles. And winker, there were no difference between light levels. We also look at plant height. We did not see any difference between plant height or any of the crops between the treatments. And then we look at plant compactness. Now, compactness is shoot dry weight divided by plant height. So higher the compactness, that means the plants are more compact, plants are shorter. So why the compactness is important? When the plants are compact, so more plants can be transported, more seedlings can be transported within the truckload, because seedlings will be transporting as several layers. If the plants are shorter, they can have multiple layers, so they can save money on transporting. We saw no difference by having higher light levels on the 2 treatments, on both of the treatment, first treatment with impatient, but the winker we saw again providing high light have more compactness, but still the treatment with 8 DLI versus 100 micromoles, we did not see any differences between 2 treatments on compactness. And then finally, we calculate the amount of energy that each treatment used. This is the energy used during the growing period by 1 light bulb. So if we're looking at the energy use from 16 mole DLI versus 12 moles, you can see 16 moles use almost twice the amount of energy compared to 12. But when you're looking at the previous data, most of the time, we did not see big differences between the growth. Therefore, for this study, we believe using DLI 12 is better, mostly in energy perspective. Even we should not go to higher DLIs, the much efficiencies we can achieve by DLI 12. And then when we comparing DLI 8, our control treatment versus 100 micromole treatments, we did not see any of the differences between these 2 treatments on previous growth data. So, but you can see how much energy being saved by having this system. So that is why it's important to have a control system on providing supplemental light. For the conclusion for my first study, the adaptive lighting control system controls LEDs precisely. It can provide the specific DLI every single day, regardless of weather. And supplemental lighting stimulates plant growth, more at higher DLIs, but we need to consider about the energy use. And finally, I can say consistent DLI will increase the reliability and predictability of greenhouse seedling production, because if you're providing same amount of light for seedlings, regardless of the weather changes, you know your plants are growing same levels at every single day. And then you can predict which days your seedlings will be ready to be harvested, ready to be shipped. Then you can, growers can have their contract to ship them earlier because they know their plants will be ready on this day. So that is the importance of this. I was planning to give you time for question. Maybe I just keep going because of the time concern. So I'm going to the second question. So the second chapter, I was trying to identify the canopy size and light use efficiency. So I've tried to identify the difference between plants. What are the factors that can determine the growth. So in this study, we did in the vertical farm, as I explained you earlier. Vertical farm has high energy cost. Just for indoor vertical farms, the energy cost can be more than 50% of the total cost. So therefore, for vertical farms, more energy efficient crops are needed. So energy efficient means crops that can produce more biomass per unit amount of energy use. So therefore, we need to screen for rapid growing crops for vertical farms. So to screen for rapid growing crops, we need to identify the basic determinants of crop growth. So the objective of this study to understand how efficiently different crops use light energy to produce biomass and determine the underlying mechanism for these differences between crops. So this study, we use Asian green Mizuna, which is a mustard leafy green, and then compare that with lettuce, because these two have two different growth rates. We saw in previous studies. And this study was done in a vertical farm. And I had 6 different treatments. They had different PPFD levels from 50 to 425. Therefore, this treatment got different DLIs from 2.9 is the minimum and went up to 24.5. And these 6 treatments were repeated in 2 other products as well in the same growth chamber. And if you're looking at the dry weight, we saw, as we expected, we saw Mizuna always had higher dry weights compared to lettuce. So we thought, we identified projected canopy size is one of the reasons to have this higher growth. So what is projected canopy size? When you provide light energy to plants, so they absorb light energy, some of them being used for photosynthesis, and some will be dissipated as heat, and some energy will be re-emitted for chlorophyll process. So this re-emitted light is emitting in red wavelength, so this is red light. So for example, if you're looking at this picture with Mizuna, you can see multiple colors on the background. So if you give a blue light to this plant, and then we take a picture with the camera by placing a bandpass filter that cannot see any of the blue light. Now we only can see red light, which is coming from the chlorophyll process. So for this method, we can easily separate background from the leaves, so canopy size. So benefit of this, we can see the plant growth throughout the growing cycle without damaging the plant by just taking these canopy images. So if you're looking at this graph shows, this figure shows graph from each individual PPFD levels. You can see all of these graph shows Mizuna had higher projected canopy size over time compared to lettuce. And if you're going to look at the last one with the highest PPFD, if you're looking at from day 0 to day 8, the small square inside, even at the very first day, Mizuna had a higher projected canopy size compared to lettuce. So this is the one way we can screen crops with faster growth. Even at the early stages, at seedling stage, earlier we can see higher projected canopy size. So we can easily screen those crops. We know those crops are the ones that grow faster compared to the other ones. And also we look at the projected canopy size at the harvest, and you can see the projected canopy size is higher in Mizuna compared to lettuce when the light level is more than 125 ppfd. And the benefits of having higher projected canopy size, so plant can incident more light because their leaves are bigger. So therefore we calculate the incident light, that's the total amount of light that one single plant received throughout the growing period. We saw incident light of Mizuna is higher compared to lettuce when the ppfd is above 125. And that is one reason that we saw faster growth in Mizuna compared to lettuce. And the second one, we saw higher light use efficiency. So light use efficiency in here is the amount of dry matter produced for mole of light product provided. So you can see Mizuna has higher light use efficiency in all the light levels compared to lettuce. So for reason for this, we saw Mizuna had higher chlorophyll content in a unit leaf area compared to lettuce. So it can absorb more light compared to lettuce. And then if you're looking at the content yield, which is the fraction of light used for photosynthesis, and higher fraction of light used by Mizuna for photosynthesis compared to lettuce. So because of these two factors, the total photosynthesis was always higher in Mizuna compared to lettuce. So that is why we see higher light use efficiency. So because of these two factors, that's why YC Misuna grow fast. So we saw my conclusions, Misuna grow much faster than lettuce, especially when ppFD is above 125. Faster growth in Misuna is one of the reasons, having larger canopy size, which can have more incident light over time, and better light use efficiency compared to lettuce. Therefore, I would like to say, understanding these basic determinants of crop growth is important when we are screening for rapid growing crops. At early stages, we can screen for rapid growing crops for vertical farms. Therefore, having rapid growing first crops, we can increase the energy efficiency in vertical farms. Study 3, good. Go 3 straight and then go for questions. So this study, the energy requirement for greenhouse lighting can be reduced by considering excess light from the previous day. So as I explained you earlier, we have a DLI guideline to grow crops in CES. We talked about seedlings earlier. Now we are going to talk about leafy greens. So leafy greens need more than 12 moles of light per day. I also mentioned the DLI varies from day to day. But in this study, we thought, can we take advantage of this natural variability of sunlight, and can we reduce the energy cost in CES for lighting? To explain our concepts, I'm showing you here 2 days, day 1 and day 2. Day 1 is a sunny day and day 2 is a cloudy day. And let's assume day 1 we get 20 moles of light from sun, and day 2 we get only 8 moles. And let's assume our plants need 15 moles of light. Therefore, on day 1, plants get enough light. And it also gets 5 moles of light extra than what plant needs. On day 2, plants only get 8. Therefore, in conventional method on lighting, we provide 7 moles of light to reach that target because we want to have the growth same. But in our concept, instead of that day 2, this is our new day 2. So we get 8 from sunlight, and we consider this 5 moles of light plant received on previous day from sun, that extra light. And then we think if we consider that, we only need to provide 2. So then 2 plus 8, 10, still 15. So if you're looking at the 20 on day 1, and we provide total of 10 on day 2, but still the average DLI between 2 days is 15. So if the average is still 15, we think the growth might be also average out. So therefore, our method versus conventional method might still give the same results. So if it does, then how much energy we can save by reducing the light? This is just for 1 day. If throughout the growing period, it's going to be a lot of saving. So therefore, we hypothesize plant might be able to tolerate lower DLI following a sunny day up to a certain level without affecting the growth. And our research goal was to identify how large DLI fluctuations can be tolerated by plant without reducing the plant growth. For this study, we did a growth chamber study followed by a greenhouse study. The growth chamber study, we use both green salad bowl and red salad bowl lettuce. And for the greenhouse study, we only use green salad bowl lettuce. For this is the 6 treatments we use in the vertical farm. So each of the treatment, we had high DLI day followed by a low DLI day. And those DLI days were alternated. For first, we had our control with 15 for both high day and low day. Therefore, the variation between 2 days was 0. And the second treatment, we increased the DLI by 2.5 on high day compared to the control. And therefore, to keep the same average of 15, we reduced the DLI by 2.5 on the low day. Now the variation is 5 between high day and low day. Therefore, likewise, we had DLI variations up to 25. The last treatment with 25, you can see high day get 27.5, low day is 2.5, but still the average is 15, but there's a huge variation between 2 days. So those 2 days were always alternated, high, low as well. And these are from the greenhouse study. You can see the DLI variations change from 0 up to 14. And the 14 is the max we were able to go in the greenhouse. So now we still have the 15 DLI average on both this, and the max DLI variation, we had 22 on high day and 8 on low day. So looking at the data from the growth chamber study, I'm showing leaf area and dry weight, red for red lettuce and green for green lettuce. So if you're looking at both leaf area and dry weight, we did not see a reduction of growth up to DLI fluctuation of 15. So when the DLI fluctuations go to 20, we saw the reduction of leaf area as well as dry weight. And when you're looking at the greenhouse study, we saw a slight reduction of leaf area and dry weight over time increasing with increasing DLI fluctuation. But when you're looking at how much leaf area and dry weight reduce with increasing the mole of DLI, increasing DLI by 1 mole, leaf area reduced by 12 square centimeters, which is a very small amount for lettuce with multiple leaves. And then dry weight only reduced by 0.02 grams for increasing of DLI fluctuation by 1 mole, which is a very small number, especially when you go to grocery store, you buy lettuce as individuals, numbers, not by weight. So this small amount is not going to be a big issue. So by comparing these two studies, we identify up to DLI 10.5, we can have DLI fluctuations for these two crops without reducing the plant growth or minimal reduction. So if we increase the DLI fluctuation more than 10.5, then we can see there's a reduction in the growth. So to show it more clearly, this is average looking plant from our greenhouse study from DLI fluctuation 3.5 up to 10.5. There were no differences. You can see 7 to 10.5 is pretty similar, and then 10.5 to 14, you can see the plant is much smaller. So we can, from this study, we can safely have DLI fluctuations up to 10.5 without reducing the plant growth. Then we use that data and we calculate the energy used for 5 different locations of the United States to see how much energy required to grow 1 acre of greenhouse lettuce production. So we kept the DLI target as 17 to calculate that. And we use typical meteorological data, that is the average weather data from last 10 years. So we use the average sunlight data for these 5 different locations and calculate the energy requirement. And this is the energy requirement without considering this our new concept. And when we consider our new concept by providing maximum DLI fluctuations of 10.5, and now this is the new energy requirement for acre of greenhouse lettuce production. And we calculate that this is into a dollar value by considering 10 cents per kilowatt hour. You can see in some regions, growers can save almost $8,000 per year, but just for 1 acre of lettuce greenhouse production, lettuce production in greenhouses. So this for growers with multiple acres of greenhouses, this is going to be a huge savings. Therefore, the conclusions of for the third study, carryover of excess light from a sunny day to an overcast day is possible, but within limits. So lettuce can tolerate DLI fluctuations up to 15 moles indoor and 10.5 moles under greenhouse condition without affecting the plant growth. Considering this method can save up to $8,000 per year on energy cost for an acre of lettuce greenhouse production. So that is the end of 3 studies. I can give maybe two minutes, take 1 or 2 questions and drink some water at the same time. I can put the caddies around me here. If you want to stop me, so it will be easy for stop. So if any burning questions, you can wait for last minute. I can answer for next 2 or 3 minutes if I move on.
I have a question about the first study.
Yes.
You indicated that the 20 days is typically the marketable point, right, for those plants?
No, I was showing the sunlight. If that is the marketable weight we need, we can achieve that 10 days earlier, 20 days,
right,
by providing.
So, but theoretically, they could be sold at 20 days, right?
Yeah, I think they were.
With the supplemental rights.
Yes,
Yeah.
that's right.
So,
yes.
do you know if they reach? I mean, there was a pretty significant increase in biomass at day 30. Do you know if that will be beyond the marketable point, or the grower could make more money?
So it depends on the crop. I don't think having a much bigger seedling growth is important, because they probably have. So I was trying to show if this is the marketable weight they need, by providing supplemental light, you can get that earlier. So it can be,
right, which is a good point.
Yeah, it can be 10 days, or it can be 15 days, can be 10 days like that. But 20 days, maybe not the marketable size. Don't know exactly. So we were trying to figure out different growth. Might be 20 days the one that marketable size. I'm not exactly sure. The 20 or 30. In 30 days, plant look much bigger. Maybe it's too late. So don't know exactly. It depends on the crop and the culture as well. Any other questions before I have? Yes.
So it would be more advantage to use the adaptive lighting control or the carryover method. Do you have like data on the comparison?
Yeah, good question. There are 2 different things. So adaptive lighting method is, I'm talking about seedlings. for seedlings, it's better to have on that study. But that also can be used for other crops as well to give the same amount of light every single day. So what we can do for when you're doing that third study with having different DLIs, to keep that DLI levels, like let's say on this day I have 5 extra lights, so next day I need 5 less. So how are we going to control that? You need the adaptive lighting control system to do that. So that is both together. Yeah, I don't explain that part because I already explained earlier. So what we did that we had the adaptive lighting control system to control that DLI program, calculate how much DLI I need by looking at the light level previous day. To really keep that light level, each do the adaptive lighting control systems, the one that actually control the light.
It's gonna control it, but different amounts each day.
Each day,
Not the same.
different amounts each day, depending on the previous day. So I'm just going to give more ones so because the time limitation. Sorry about that.
I have a question, TC.
Okay, last question. Yeah.
Yeah, this is Tessa. Really great talk so far. I have a question about the first study. And, you know, the yield did not change that much when the DLI was increased, right?
Yes.
But the kilowatt hours did. Have you calculated grams per kilowatt hour at all?
I have not, but I have that graph. I'm working on it. I'm trying to, when I publish, I'm going to do that, but I haven't calculated at the moment. I'm trying to calculate gram of root mass production, especially for kilowatt hours, because root is much important. I haven't done that yet.
Great. I look forward to that.
Okay,
Thank you.
All right, go to the chapter 4. Hydroponic lens production can be accelerated by providing more nitrogen at high light levels. So growers try to put more light to their plants because they can sell their plants quicker than without low light levels. So having more light levels is more important for them, providing more light levels. But what about nitrogen? In there's higher light levels increase as increasing photosynthesis. Those plants need more nitrogen as well. So what is the importance of providing more nitrogen? So if you're looking at the photosystem, there are mainly 2 stages: light reaction and the Kelvin cycle. So if you're looking at the light reaction, so if when you're providing more light, plants need more chlorophyll to absorb that light. So nitrogen is a component of chlorophyll. Therefore, more nitrogen need to provide more nitrogen need to synthesize more chlorophyll. And if you're looking at the Kelvin cycle, Rubisco is one of the main enzymes in Kelvin cycle. To speed up the Kelvin cycle at high light levels, plants need more Rubisco. So Rubisco is an enzyme. So enzyme is made of amino acids. Nitrogen is one of the key components on amino acids. So to produce more Rubisco, more nitrogen is required. Therefore, at high light level, to increase the photosynthesis, more nitrogen is required. If farmers trying to give more light, why they don't put a little bit of more nitrogen so they can get more benefit out of their amount of energy they use. For objective of this study to determine whether additional nitrogen is needed for promoting faster growth crop under high light conditions. This study was done in the greenhouse in hydroponics with butterhead lettuce rex cultivar. And as you see in the picture, we use 9 greenhouse benches. Each bench has 2 trays, and each of these trays has 36 plants. And as you see in the diagram on the right, so we had 6 different blocks. So each block has 3 trays. So you can see the letters A, B, and C on each of these trays. Each block has A, B, and C. A, B, and C represent different nitrogen levels. Excuse me, A for 100, 200, and 300 for C, milligram per liter nitrogen. And we use Hort America's mixture to make that 100%, 100 milligram per liter A solution, fertilizer solution, and then we use ammonium nitrate to increase the nitrogen of the other 2 solutions. And as you see in the picture, the light is located at the center of the bench. To see it much clearly, this is a diagram of one bench with 2 trays. So this is where the light is located. So because of the location of the light, each of the plant get different light levels. So therefore, we determine using a contact sensor how much light each plant get at when the lights are at full power. By looking at these numbers, we categorize the total plant into 6 different groups by putting the plants with most similar light levels together. So with that, that's why you can see A is at the center because it's right underneath the light. And if you go from B, C, D, E, and F, F is the furthest away from the light, has the lower light levels of 179 micromoles, and the highest of A was 468. So these are our 6 different lighting treatments. And then we also have 3 different night gen treatments. So we expected to see more photosynthesis with increasing night gen, but unfortunately we did not see any higher CO2 assimilation or higher photosynthesis with changing night gen levels. For reason for that, we think one of them, as you see in the chlorophyll content, when the night gen level is 100, which is in green, you can see the chlorophyll level is lower compared to the treatment with higher night gen levels. So those plants can absorb less amount of light compared to the other plants. But if you're looking at the quantum yield, which shows the fraction of that absorbed light used for photosynthesis, when the night gen level is lower, those lights is used more efficiently. That's why you see higher efficient light used when the night gen is lower. Because of these contrast factors having low absorption but used them efficiently at low night gen compared to high night gen, that's why you probably didn't see a difference between photosynthesis level. And then the growers don't matter about photosynthesis as long as the shoot fresh weight is grown, if they are later. So we saw increase of fresh weight with increasing night gen levels. And you can see the difference is higher at higher light levels. And also we saw that pattern very clearly when you're looking at the shoot dry weight, higher the night gen at all the light levels, dry weight was higher with higher night gen. But the problem is tip burn. So tip burn is what when the fast growing tissue does not get enough calcium to build up cell walls. Therefore, you can see the burning symptoms from the edge of the fast growing tissues over here at the center. So we saw tip burn symptoms increase with light levels, we know that, but also night gen levels, when providing more night gen, tip burn symptoms increased. For reason for that, we show shoot dry weight increase with, we know shoot dry weight increase with night gen, we saw it earlier. When we increase the shoot dry weight, the tip burn increase. That's why we saw higher night gen levels have more tip burn symptoms. And the reason for tip burn is having not enough calcium. We look at leaf calcium concentration and leaf calcium content. So leaf calcium content, the graph D, that it shows the amount of calcium, total amount of calcium that one plant has. And the concentration source show that calcium, amount of gram of calcium within the unit dry weight of leaf area. That's the concentration and that's the content. So if you're looking at the content, there's no difference between different night gen levels. So if you grow the plants at 100, 200, or 300, they all have the same amount of calcium. But what happens when you have in high night gen, they grow much faster. So they produce more shoots. Because of that, the concentration goes down. Therefore, at higher night gen level, low concentration at shoots, that's why we see tip burn patterns happening at more tip burn happening at higher night gen levels. So as conclusion for this study, adding nitrogen is favorable for promoting faster lettuce growth under high light conditions. So faster growth can promote tip burn symptoms. So therefore, to reduce the tip burn symptoms, we have to increase the canopy airflow so plants can absorb more calcium, or we have to reduce the cultivars. There are some cultivars are much tip burn resistant compared to the other cultivar. So crops that can be used to get this benefit of providing more nitrogen at higher light levels. And just keep going on the last study. This is the supplemental far-red light study. So I'll be done after this. So supplemental far-red light does not increase growth of greenhouse grown lettuce. So as I explained you earlier, the far-red range from 400 to 700, and the far-red light is from 700 to about 780, just below the infrared rays. So together these lights called EPA, extended photosynthesis active radiation, because there are several studies done in our lab and many other labs. If they are seen, indoor studies seen adding far-red light to far-red increase the plant growth, plant can grow faster. But those studies are done in indoors, not in greenhouses. So why this benefits of far-red light? By providing far-red light, what is this benefit for plants? So if you're looking at the photosynthesis light reaction, there are two photosystems, photosystem 2 and photosystem 1. If you know about the photosynthesis, I'm pretty sure most of you know. So photosystem 1 is preferentially excited by far red light. Therefore, it restores the balance between photosystem 2 and photosystem 1 to increase the efficiency of electron transport rate. By increasing the efficiency of electron transport, it can increase the photosynthesis efficiency. And the second thing, if you're looking at this crop, the huge plant, under plant, so when the plants get sunlight, most of the red light being absorbed by plants. Therefore, the red to far red ratio reduced under plant canopy. That means more far red under plant canopy. So that triggers the morphology of crop, which regulate the plant morphology. So plants try to get more light by providing more far red, this triggers, and then plants can either go up or they can expand the leaves so they can capture more light. So that is another benefit of having far red light. So we try to mimic this condition by providing far red, then plant thinks, okay, we need to expand the leaves or increase the height. So most of the leafy greens, including lettuce, expand the leaves. So that we take the benefits of that by providing far red light. So this is a study, indoor study done in Michigan State, showing on the left the leaves, seedlings, lettuce seedlings without far red and with far red. You can clearly see the leaf size. If you're looking at the leaf size, they are much bigger with providing more far red. Why is important to have bigger leaf at early on? So they can intercept more light, they can get more orange light, and then those crops can go faster. Those seedlings can go faster compared to the seedling without far red. So our objective is to quantify the effect of supplemental LED light with far red light on greenhouse grown lettuce, because all the studies done, most of the studies done in indoor farms. It's very hard to see in a study done in greenhouse to see the effect of far red light. So over here for this study, we use green salad board and a red lettuce Cherokee, and we use a Lumigro lighting fixture. And this lighting fixture has a light. You can see we can control red lights versus blue light and white light. By controlling, you can change the spectrum of the light. With that, you can change the color of the whole greenhouse. That was so fun. So I was driving by greenhouse that time. I just changed the color of the greenhouse with my phone into red, blue, purple, or if it's boring, I just change it to white, like that. So that light is so cool and it has so much fun. But for this study, we kept 3:1 ratios of red to blue. So Mark didn't let me change it. And then in addition to that, this light didn't have any far red light. So we added custom-made far red light bars, which these light bars are super boring because you cannot change any spectrum, and it has far range range at the end of the visible spectrum. You cannot even see that light from your eyes. So that's why I call this. One reason I call this light are boring. And we added our treatments with far red and far range. We provided light 4 hours before sunlight and 4 hours after sunset, so we can get the maximum benefits of light spectrum, changing spectrum when there's no sunlight. And so talking about the treatments, we had 5 different treatments. First one is PPFD 200, just providing by Lumigro light with far range without far red, and then 200 plus 10 far red, and 200 plus 20 far red, and the fourth one, PPFD 220, because if we saw any benefits of adding 20 far red on the third treatment, we want to know whether it's because of far red or it's because of adding just 20 moles of extra light. So we kept the fourth one as 220 without far red, just from far red. And the last one was sunlight only. On average, we get DLI about 7.5 from sunlight, and from the PPFD light from 200, we get 5.76 of DLI, and it was 6.34 on average from PPFD 220, and we get 0.29 and 0.58 moles of light from far red light on 2-day treatments. So we're looking at the CO2 assimilation photosynthesis. As we've seen in the vertical farm, we did not see any differences on any of the treatment by providing far red light. So that is the second reason I'm telling these lights are boring, especially in the greenhouse condition. There's no difference in CO2 assimilation. And if you're looking at the dry red, there were no differences as we needed. We saw the far red going to have more difference, more growth, but we saw a slight reduction in adding far red light. But there were no benefits of adding far red light in greenhouse condition. But generally, providing any supplemental lighting in the greenhouse, we were able to get 151% growth in green salad bowl and Cherokee 101% growth. So that growth was taken by providing 82% of more light. So providing 82% of more supplemental light we can get 100 to 150 growth. So that's the benefit of providing supplementary light, but nothing related to far-red light. So therefore, my conclusions, supplemental far-red light has no additional benefits on growth of greenhouse grown crops. We did this study like a year and a half. I did 3 studies, lettuce with cilantro, with lettuce, with basil. We did not see any differences. And then supplement, however, the supplemental far light in greenhouse increased the crop growth. And we believe plant may receive adequate far-red light from sun as compared to the indoor settings. Indoor, there's no far-red light. That's why we did not see any benefits of adding far-red light in the greenhouse. So even these studies has no good results. We think it's better to tell people, don't just try to waste your time on trying to find effect on far-red on greenhouse, just like what I did. So before we go to questions, my broad objective, I want to remind you again, exploring ways to optimize the use of horticultural lighting in CEAs, control environmental agriculture, to achieve a greater energy efficiency and more productivity. For my overall conclusions from the first study, the adaptive lighting control system can control LEDs precisely to provide a consistent DL light, which help to increase the reliability and predictability of greenhouse seedling production. And the second study, so determining the canopy size and light efficiency is important when screening for rapidly growing crops, which help to increase the energy efficiency in vertical farms. And the third study, considering the excess sunlight from a previous sunny day to overcast day, can save up to $8,000 per year on energy cost on some areas of the United States per acre of greenhouse lettuce production. And the fourth study, providing more nitrogen, promote faster lettuce growth under high light condition. And the fifth one, you can read this if you need by just saying, don't waste your time trying to find effects on far-red light in greenhouse. With that, I would like to thank Mark Van Eschel for helping me throughout this process and all the guidelines he's given me through the last 5 to 6 years. And also my committee, Dr. Ronito, Tim Kolo, Mark Heidecker, and Tessa Pocock. Thanks for joining today as well for all the guidelines given. And finally, I would like to thank all my lab mates. I tried to put all the pictures, everybody, from my former lab mates, the current lab mates, all the student workers, visiting students who helped me during these projects. Thanks to you, I'm presenting this today. Without you, I'm probably still in the greenhouse harvesting and collecting data. Thank you very much. And funding sources, and then any questions if you have any.
Questions?
We know we're having a disco in your greenhouse.
Did you see that?
I have a question. Tip burn is rather close to my heart, in industry on cilantro lettuce. Do you think, TC, that if we reduced our nitrogen levels, I mean, we grow at moderate light, around 450 micromoles. Do you think if we reduce the nitrogen, we could reduce the frequency of tip burn in our crops?
So we saw higher nitrogen provide higher tip burn. So I believe reduced nitrogen may have reduced tip burn as well. But it's completely different on the type of the crop we always saw. Different crop might need different nitrogen levels.
You've given me a good idea. I think I'm going to try that and see if it helps. This is great.
Any other questions? Yeah.
Did you ever think about using calcium nitrate to like add calcium and more nitrogen to your elevated nitrogen levels? Like to
Yeah, we can.
tip burn?
So when you're looking at the calcium content concentration, so yeah, providing calcium nitrate, yeah, that can be other alternative providing more calcium. But the thing is, as you've seen, plant has enough calcium. There's no difference between different nitrogen levels. But the dilution is the problem. So with the growth. So how are we going to give more calcium into that fast growing tissues at the center? Because calcium is coming with the transpiration. Most transpiration happening in the bigger leaves on the side, not the center. So less transpiration at the center. Therefore, more calcium goes to the outer leaves. That's what I believe. So that's why we see how low calcium goes to the center leaf. So if we can increase the airflow there, then we will have more transpiration. Then we can send more calcium will go to these faster growing tissues and that can help the reduction of timber. But for my study, I could not use calcium nitrate because then there will be two factors of providing calcium and nitrogen. So I had to keep only ammonium nitrate, which only give nitrogen. Yes.
So do you think, I mean, I'm not that familiar with Tibbin, but it just, everything you described with faster growth and calcium just seems like the kind of the same thing as blossom end rot we get in fruit development.
Yeah.
And so I was wondering whether those two things are related at all, because that would
Yeah,
be really interesting.
I haven't done any study with food, but as I understand, that is also a tick-borne, so calcium deficiency symptom. But I don't know how to address that because foods, maybe there's fruit don't get enough calcium as well, just like lettuce. But I don't know whether transpiration like increasing airflow will going to be helpful for fruits. Because I don't know whether they're going to reach transpiration, so I don't know how to do. Maybe foliar spray on fruits or something like that. So it probably different approach how to do on fruit compared to leafy vegetables.
We got Savitri, they do work on that, but yeah, that's pretty interesting because we see it in longer fruits. So when they
Yeah, especially bell peppers has too. Right?
yeah,
I've seen that.
watermelon also,
Watermelon,
and the elongated watermelon, yeah, and pepper, tomatoes. So you see it in.
Did you try any calcium spraying? Whether that going to help? Or I'm not sure.
So I mean, people do give extra calcium, but it's not so clear cut that that totally
Yeah.
solves the problem.
Any other questions? Oh, yeah, yes.
Yeah, I have a question like when you are providing high DLI, then the lettuce have less yield. Why it's happen like when you're
Let us have less yield.
Yeah, like it has tip burn or some kind of
For the ideal, I always increase the yield, but the tip burn is the problem because the higher light.
usually we say like when you are providing high DLI, then it gives the high yield.
Yeah.
But in lettuce, in your case, in your first slide, you mentioned like when you're providing high DLI, then it decrease the yield or it has more tip burn. Right, something like what is
So the reason for DLI, when you're providing high DLI, so there's a, lettuce has a range of DLI as I showed you earlier. Usually for 16 to 17 is the DLI recommended for lettuce. Mostly what happens when you give more DLI, that means you're providing more amount of light within a day. So those crops achieve growing degrees faster, much faster than having low DLI. So having more growing degrees, that means it's getting more light and the growth is much faster. So then plant probably cannot stand with that faster growth. So especially high DLI, as I mentioned earlier, the faster tissue growth. So faster tissue growth, and also, like for example, for lettuce, at the middle is the like younger leaves. So younger leaves grown faster at the center. Those plants don't get enough light, enough calcium. So higher light, plants try to grow faster, but if they don't have tip burn, that's better. But the problem is we need to provide a way to give enough calcium for these fast growing tissues. Then we can get the benefit of higher light. Is that the question you asked?
Yeah, that's true. So there are also some kind of light suppress here, but we
Some kind of a light effect.
light suppress on the plant.
Stress.
Yeah.
There can be some stress as well, but we did not see any other stress other than these light levels. But if you go DLI further high, there can be a photo damage happening to plants or something like that because of having more light levels. Any other questions?
Regarding your far red light experiments, did you keep track of anthocyanin content in red cultivars of lettuce?
I probably did not. That was, yeah, that was done really early. Maybe I did several studies, like 3 studies or so. So some studies I only had basil and cilantro. I did not do any anthocyanin. Probably the study I showed you, maybe I did anthocyanin, but I did not look at the data to see what's the difference between different treatments like that. So one thing, anthocyanin, anthocyanin just have less light coming into the plant leaves. So having far red, maybe I don't know what will happen on cap. So I saw in the picture that as I show you far red, light has more anthocyanin production with less violet compared to violet, because I think they might have the same anthocyanin, but the leaves are growing faster than the anthocyanin is distributed among more leaf area, so you see less color within the unit area, so that can happen, having more violet, more growth. I saw someone else. Yes.
Okay, on your second study conclusion, so you were comparing two crops of lettuce and a lot of plants, and you say that like providing more light, like more than 125 PhD, which that's when you see a difference. And why not earlier? What's happening below that PhD?
So because as I showed you earlier, the projected canopside, we did not see. So Misuna at the lower light level, we were doing it really poorly. So they're like really high light levels. So they had really low projected canopsize compared to lettuce at low light level like ppmd 50. So because of having low light levels, they absorb less light compared to lettuce at lower light levels. So when the ppmd is more than 125, we see the benefits. And usually for lettuce, on most of our studies too, we provide 200 to 250 to 300 ppmd. So at that level, we should not do much better compared to lettuce. Projected canopy size, the canopy size is the problem. You should ask me that, yes.
Yeah, like, I mean, like, yeah, I see, but what, do you know the reason?
For having no
Yeah,
projected canopy size?
No, like for the Misuna to not do well. Like,
Because of not having enough bigger canopy. They did not like low light levels. They like high light. Any other questions? Anything online?
Well, if there are no more questions, then TC, thank you very much for this great seminar. I'll give you a virtual hand.